Hi everyone, and welcome to our video, part of our Module 5 series, the neuronal communication. And what we're going to be doing here is having a look at the structure and function of neurons. So the particular specification reference that we're looking at is 5.3.1b, the structure and function of sensory, relay and motor neurons. One of the aspects on the specification we need to understand is not only about the structure of these neurons, but also their particular functions. Now, the general function of a neuron is to transmit the impulse that was generated from that depolarization of the receptor cell membrane that we looked at in the previous video to other regions of the body. And we mentioned this phrase action potential in our last video, and that's the method through which this is going to be transmitted. So being back at GCSE, we would have looked at these three types of neuron: sensory, relay and motor. When we're talking about the sensory neuron, they are the ones that will carry the action potential from the sensory receptors to your central nervous system. The relay neurons are the ones that carry out that connection between the sensory and the motor neurons. And finally, the motor neurons themselves will carry that action potential from the central nervous system to one of the effectors, which are the muscles and the glands. If we now start to think about the structure of these neurons, there are a few key features that you should bear in mind. So the first one is that they have a cell body which contains the nucleus. And obviously the nucleus is where we've got our genetic material. In addition to that nucleus, we also have many mitochondria and also ribosomes present. They have this structure called an axon, which is going to carry those impulses away from the cell body. And what we find is that many of these neurons are actually really quite long, because they're trying to cover very large distances within the body of the organism. They can also be surrounded by this fatty layer, which is going to insulate the cell itself, and we'll look more at that in a later part of the videos. And they've got lots of these structures called dendrites, which are the connections to the other neurons. So when you look at the pictures, they're the kind of bits that stick out like little fingers or roots or branches at the end. Three other bits that we also need to know is that they've got a cell surface membrane with many gated ion channels. And these gated ion channels are going to be for three particular ions: sodium, potassium, and calcium. So we've got these different gated ion channels for those three key ions. We also find another structure to do with our ions, the sodium-potassium pump, which is all to do with active transport of the sodium ions out of the cell and the potassium ions into the cell, as we saw in our previous video. And they're going to have this potential difference maintained across their plasma membranes. What we're going to do then is have a look at each of those neurons in turn so that we'll be able to identify them should they come up as questions on our exam. So first one is the motor neuron. Now, motor neurons are quite straightforward to spot because what you're going to be looking for is the location of the cell body. Now in this case, what we can see is the cell body is at one end, so this structure here is the whole cell body. And what we can see is in this central part of it, we've got our nucleus, and then we've got these little branches sticking off, and these little bits at the end here are the things we call dendrites. Now the cell body itself is then connected onto the long strand here, which is the axon, remember, and then at the far end what we have down here are going to be the synapses, so the connections that are going to other neurons. So when it comes to the motor neuron, what we're going to find is the cell body is at one end, not in the middle at all. Now, the cell body itself will be located within the central nervous system, and this one typically has a long axon. Our second neuron then is the sensory neuron. So if we start by looking at the diagram at the bottom, I always think this one looks kind of like a weird little alien, because what we have is in the middle we've got our cell body with the nucleus, which looks kind of like a little eye really, and then what we have at the two ends are these branching like structures that almost look like weird little tentacles. So our sensory neuron has the cell body in the center roughly, And then we are going to have branching bits at both ends. So on one end, we are going to have our dendrites, and the dendrites connect on to this region called the dendron. Then on the other end, we've got our little axon terminals, which are going to obviously have a synapse to connect to something else, and then they connect to the axon. So what we actually find is the cell body itself is located just outside the central nervous system. They do have this long dendron from the sensory receptors. So what we'd have over here are our little receptors. They would then obviously transmit that action potential through the dendron itself. And then we have the short axon to the central nervous system. So obviously our cell body located just outside the CNS, and then the short axon to the central nervous system. Third and final one is the relay neuron, and this one is the one that stands out. It's the easiest one to spot in a diagram and therefore the least likely to come up on your exams, let's be honest. So when we're looking at a relay neuron, it kind of looks like a little star shape really. So in the center we've got our nucleus, we've got the cell body, and then we've got this little axon, typically, sometimes it's a bit longer, but the axon then comes off. But generally is the one that looks kind of like a little star with potentially one longer little leg sticking off it. Now, one thing that we do have are the dendrites, and they're actually large in number, but they're very short. And then we do have that axon varying in length, leading to the axon terminals, where it will have a connection to something else, another neuron. If we now consider the potential questions that can come up to do with our different neurons, we've just seen that two of them are actually quite similar in a number of ways, and therefore comparison question makes sense. A comparison between the sensory and the motor neurons. So if we consider the similarities between those two structures first of all, they both have those dendrites, they both have an axon, they both have that cell body which contains the nucleus, they are both myelinated, and you would have seen on the diagrams that along the axon you've got these little blobby bits sticking off the outside. So that's the myelin, and we'll be looking at that in more detail later on. And they both have sodium potassium ion pumps or voltage gated channels present in their structure. We've mentioned this thing called myelin, so let's delve a little bit deeper into finding out what this myelin thing is and what it does first of all. So what we actually find is that if we examine the sensory and motor neurons of most organisms, then the vast majority of them are actually going to be myelinated. So they will have this myelin sheath around their axons. And basically what this is, we've got our neuron, and then we've got these things called Schwann cells, which are basically lipids, wrapped really tightly around it. Now, this isn't a continuous covering. What we actually find is that every 1 to 3 millimeters, there's actually going to be a short gap. Now, we are talking living things, and obviously as we go into living things, we're talking cells, and cells aren't actually big in a number of cases. So when we're talking about a short gap, we're not talking about one that you could see with the naked eye. These are 2 to 3 micrometers in size. So basically we've got these little blocks, and between them this teeny tiny little gap. And those little tiny gaps in that myelin sheath are called the nodes of Ranvier. In terms of why this myelin sheath is actually important for organisms then, is first of all, it's going to prevent the movement of ions across those neuron membranes. Now, what that means is we're going to have a big restriction on where the ions can actually cross those membranes. Because if you remember, what we've just said is that myelin covers a large proportion of the actual neuron itself, with only these teeny tiny little gaps every so often. So it's only at those nodes of Ranvier, those little tiny gaps, that the ion movement can actually take place. So what this actually means in the grand scheme of neurons and their function is that the only way that we can generate this action potential is if ions can cross the membrane. But the only place that those ions can cross the membrane is at the nodes of Ranvier, where we've got the gap in the myelin. So we can actually state that the action potentials can only be generated at the nodes of Ranvier. And what this actually does, and I know it sounds a bit weird when I say this, but it speeds up the transmission of that nervous impulse through our neuron. And we'll look and see how it does so in a moment. The non-myelinated neurons, though, are the ones that are going to be associated with those Schwann cells, but they're quite loosely wrapped. So we don't have that very tight wrapping that we saw with the myelinated neurons. Schwann cells are there, but they're quite loose in their wrapping. And therefore, what we find is that these non-myelinated neurons are going to have these action potentials moving as a wave. They can't just jump from one of those nodes to the next because they don't have them. So that loose wrapping means that we're going to have the ability to have ions crossing our membrane throughout the structure, and therefore transmission through these non-myelinated neurons is slower. If they're myelinated, it's quicker because to all intents and purposes, it's jumping from one node to the next. So let's do our comparison, because again, compare two structures is always a good question on an A-level paper. And we're going to compare the myelinated versus the non-myelinated neurons here. So three aspects we'll have a little look at: the speed, the length, and their potential uses in the organism's body. So first one then, our myelinated neuron is the one that's going to be travelling fast. Okay, so this has the rapid transmission because the action potentials are basically jumping one node to the next. And what we find is the actual speed of these is between 100 and 120 meters per second. So that's pretty darn quick. Whereas the non-myelinated ones, these ones where they've got to travel as that wave, it's going to slow right down to between 2 and 20 meters per second. Now, it's a good point just to stop and point out how we actually use proper scientific notation for units at A-level. So I'm sure that when you were down in GCSE, you were doing all this M/S stuff. Please don't on A-level. Let's actually make sure we're using correct scientific notation. That is a no-no. Let's make sure we're using meters, second, the power -1. So meters per second, correctly written. Now, in terms of the length of the neuron, what we find, the myelinated ones tend to be longer and the non-myelinated will be shorter. And their typical uses for the myelinated, these are going to be the ones we're utilising when we're responding to a stimulus. So this is something where we need the body to carry out some kind of a response quickly, typically to avoid further injury or harm. Whereas the non-myelinated ones, because they have this much slower speed of transmission, they tend not to be associated with those kind of protective features of the body. They're instead going to be involved in bits where speed isn't so important. So coordinating those basic body functions like breathing. We don't generally need this very rapid transmission of information to control that. It's a more steady process. So anything that requires a very rapid response, probably going to be a myelinated neuron because they've got the faster speed of transmission, because with that myelin sheath, it's going to have the action potential jumping from one node of Ranvier to the next. Non-myelinated neurons, slower speed of transmission for coordinating those body functions where speed is not of the essence. As always, I do hope you found this video useful, and don't forget to subscribe to the channel so you can see when another video is uploaded, and of course head on over to the website where you can find a range of other resources to do with your A-level biology course.